Hello, welcome to the Trailblazer Research Seminar Series, a production of the NUS MedSci Alliance. This video was recorded on the 29th January 2026 at NUS, with our speaker, Professor Prof Clemmensen, sharing his research journey about mechanism of weight homeostasis and the future of obesity pharmacology. Let's tune in and see what he has to say.
Hi, I'm Prof Clemmensen from the University of Copenhagen, and I'll be giving a talk on mechanisms of weight homeostasis and the future of obesity pharmacology.
Hello, everyone. A very good afternoon. And thank you all for being here today. And hope 2026 has been a good start, right? So this is our Trailblazer Seminar Series of 2026. And it is our great honor today to have Professor Chris Clemmensen from University of Copenhagen to join us. So a very quick introduction of Professor Clemmensen before we invite him to on the stage. So also a quick introduction of this Trailblazer seminar organization. So there will be a 30 to 35 minutes talk. Then we will follow by question and answer from the audience, some fire chat discussion. So I would strongly encourage the junior researchers to engage with the speaker during the fire chat, also after the seminar, during lunch. And so our Professor Clemmensen is now associate professor and group leader at the Novo Nordisk Foundation Center for Basic Metabolic Research, University of Copenhagen. He was trained at the University of Copenhagen with Bachelor of Science in Exercise Biology, Master of Science in Human Biology, and a PhD in Molecular Pharmacology. He then pursued a postdoc training in Helmholtz Diabetes Center in Munich and become a group leader there before returning to Copenhagen. Very excitingly, he's now the co-founder and CSO of the Ousia Pharma Alps, a biotech venture developing the novel peptide drug conjugate therapies for obesity and brain disorders. So I know some of you here are very interested in both metabolic, obesity, and brain disorders. So today is our really a great honor to have him here to share his science and journey with us. And we want to mention, there are so many awards he has gone, and very importantly is he actually is now leading a European Research Council Consolidator Grant to advance his research in metabolic health. And he recently, his Nature paper, which is very well known, that is right now is trying to provide a new therapeutic possibility for obesity and maybe many other disorders. So without further ado, let's invite Professor Clemmensen to get on stage to share with us the science and research journey.
Okay, this is better. Yeah, good afternoon. And before I start talking about our work, I just want to really thank Helen and everyone for inviting me and welcoming me and kind of organizing such a very exciting program, meeting many researchers from here in translational medicine, basic researchers, translational researchers. It's really exciting to be here and learn what you're working on. So I'm really honored to get the opportunity to also be able to talk a little bit about the work we're currently doing in context of basically weight homeostasis. So what are the biological mechanisms that regulate body weight? What are the pathological mechanisms that leading someone to develop excess fat mass and obesity? And then what is the current state of the art when we talk about drugs for treatment of obesity? And what does the future look like in terms of obesity treatment? Those are some of the topics I'll try to cover, and I'll try to do it in 30 minutes or so, and then I really invite a discussion because these are topics that I think could be very interesting to also get your perspective on. So when we talk about weight homeostasis or energy homeostasis, there's different ways to kind of go about this, but I think one way that we like to think about this is that we place our genome and especially our central nervous system is being bombarded with a ton of external input from the modern environment, right? And somehow this has to be dealt with in terms of behavioral consequences. And when we talk about energy homeostasis or balance, those consequences are typically eating behavior and food consumption, energy expenditure, or how we deposit the fuel we get into our system, right? And of course, many of these items that are depicted over here are relatively new when we think about evolutionary aspects for which our human genetics have been exposed to, right? So many of these kind of items that we are now kind of exposing ourselves to in modern life are relatively new. Irrespective of the consequences, we see a wide variability in terms of body weight or body mass index or fat mass. And we look at modern societies, and there's large geographical differences also. It matters actually quite a bit whether you live in certain aspects of Asia versus Europe versus the US in terms of the general population and its body size. Another way of depicting this in kind of a cartoon fashion is to emphasize the same point. Basically, I think if we talk about the evolutionary history of Homo sapiens, it's kind of believed that the modern humans evolved 300,000 or so years ago, right? So it's only within very, very recent kind of millisecond of time in our evolutionary history that we have been living in these quote-unquote obesogenic environments. So most of our time as a species, we've been living in relatively calorie-restricted environments, meaning that if you were carrying a genetic sensitivity to develop excess body weight or fat mass, you wouldn't have had the environment to unleash that genetic potential, right? So it's only in context of the modern environment, the last 50 or so years, we've seen kind of the full, maybe not the full, but we've seen genetic susceptibility and the spectrum of genetic sensitivity, individual sensitivity, being exposed to modern environment, and we see some people develop excess body fat mass in modern environments, right? So I think this is something to keep in mind also when we kind of talk about this topic as a whole. So the consequence is that today it's estimated that around 1 billion individuals on this planet live with a body mass index, which is kind of one way of classifying body weight variability or BMI. So body mass index above 30, meaning they're kind of classified as being obese. So it's quite large numbers, right? So why is it so difficult to lose weight? So many public health authorities, especially at least in Denmark, where I'm from, have been telling the population for many decades, you should eat healthier and you should exercise more. So be active and then kind of try and cut down on your calories, and then we'll solve everything, right? So, but it seems as if those kind of well-meaning advice have had very little impact on obesity rates that continue to rise, right? And it's likely because, and this is simplistic, right? So body fat mass is very fiercely defended by the organism, especially by the central nervous system. So somehow, or for some reason, once the body has acquired excess energy in terms of fat stores, it's kind of willing to protect that excess energy with all of its biological powers. So it's really fighting against very strong biological forces when you try to lower your body weight through dieting or other means. So another way of depicting this is that any type of weight loss intervention, could be diet, exercise, or pharmacology to try and lower body fat mass, is typically being countered by substantial neuroendocrine feedback, and the consequences have been estimated to be a decrease in energy expenditure for every kilogram you go below your kind of individual target weight. You'll see a decrease in energy expenditure by approximately 25 kilocalories. But I think what's more kind of difficult to combat is the potential increase in appetite you see. So if your genetic target weight in the environment you're in is whatever kilogram of body weight, you go 5 kilograms below that, you'd see a disproportionate decline of energy expenditure by 125 kilocalories, and you'd see a push towards over-consuming 500 kilocalories or so to re-establish body weight. And this is what we see in more or less all studies where we follow individuals for two years or beyond, that weight will eventually rebound. There's many signals involved, and of course, I think a central signal in the neuroendocrine feedback in context of a negative energy balance is the adipose hormone leptin, but there are of course many other signals involved in this process, but it's clear that leptin plays a key role in context of negative energy balance. So lower levels of circulating leptin is a very powerful homeostatic regulator. So there are many examples, and I'm not going to go too much through this because I also want to go and talk about the obesity pharmacotherapies. But I think this is kind of an illustrative example, right? So 20 years or so, these kind of TV programs were popular in the US. We didn't have them in Denmark, but we have other ones. But these type of Biggest Loser programs with, of course, they were televised, which also has some aspects, but 30 weeks of intense lifestyle intervention, you see a decrease of, average decrease of 60 kilograms body weight, right? And then they regain most, but not all of it. And there's a lot of individual variability. But I think what's key here is that they regain back to 132 kilograms. So it again emphasizes the point is that if you have acquired excess body fat mass and the body is kind of sees this as your kind of target weight, then it's actually willing to go back to that point, although it being pathologically very high and really detrimental for your health, right? So it shows again, although these are down to 100 kilogram or so in average, it's still the body pushes us back to 130 on average. So it's really strong homeostatic response. There's done thousands of animal studies. This is just an old rat study I found in the literature. Basically, just show a weight curve of rats in a given environment on this rat chow diet, right? But again, it's clear that if you calorie restrict these animals, they will of course lose weight. And once you release them again, they move back. Many of you are kind of aware of these experiments. I think what's fascinating is that they seem to go back to the same trajectory. So it seems as if you put any animal in a certain environment, right, you see almost like a programmed body weight trajectory, and somehow it's encoded in the animal, right? So if you restrict it or if you force feed it, you'll see that it goes back to this kind of programmed weight trajectory. And then moving a little bit more into kind of contemporary research. So this is something that we're now also seeing with the modern weight loss therapeutics, right? So probably the most well-known drug today is semaglutide, which is marketed as Ozempic for diabetes or Wegovy for obesity. And the studies are quite clear, right? You see a quite dramatic lowering of body weight over 68 weeks. But once you stop treatment, right, you see this very, very powerful weight regain back towards baseline. Again, emphasizing the same homeostatic feedback forces at play. So one thing that we've been very interested in my lab, and I think that's kind of less appreciated, is that actually this homeostatic correction of body weight goes in both directions. So the first studies, and I think they're quite well, kind of sometimes somewhat well known in this field, are the overfeeding trials in the late 60s by researchers from the US, where they took prisoners, inmates from the Vermont State Prison, and then they overfed them. So they motivated them by giving them they got some financial support in the prison if they kind of would eat more food than their body would need, right? And then they could see that some of these prisoners gained 20 kilograms or so over a period. But then once they released them again from the overfeeding, they actually lost the weight again. So there are circumstances where you can gain excess fat mass and then the body is willing to lose it again. And maybe we come back to why that perhaps is. There's also other exciting examples. This is from northern Cameroon, a tribe in Africa that have kind of fattening session amongst young male before marriage. So I don't know if it's because they kind of need to kind of acquire more energy before they enter marriage, but that's a part of the cultural aspects, right? And then over a period of 5 to 6 months, you see a weight gain of 17 kilogram in this fattening session, 12 of which are body fat mass. And then again, if you go back 3 years later, you see that they've beautifully recovered back to baseline. Again, showing this homeostatic correction or this kind of programming of body weight goes in both directions. So this kind of brings us to a model of weight regulation that I think we use a lot in the lab or talk about a lot. Now I see if I get the signal back here. Okay, maybe I touched something I shouldn't have. Okay, here we go. No stress. So many textbooks, we talk about like body weight set point, and it's kind of inspired by other homeostatic systems such as regulation of blood pressure or blood sugar or water-salt balance. Same could be said perhaps about body weight regulation in terms of these homeostatic regulators. But I think it's clear also that many individuals experience that they actually can vary over season or year, kind of 2 to 5 kilograms without really being fiercely protected around a very set point. So kind of one model that has been kind of introduced in this context is this dual intervention point model, where there's a lower intervention point and an upper intervention point. And between these intervention points, we have less of this homeostatic feedback forces at play, right? But once you start to deviate, you go on a diet and fat mass becomes too low, the body starts to combat this. And the same in the other direction with the overfeeding trials, we also see a correction, right? And this model has been largely popularized by a scientist called John Speakman, but there has also been kind of introduced a little bit earlier in the literature. So the reason why I like to kind of introduce this also, it kind of fits quite well in terms of the two main research themes in our lab, and that is we try to kind of come up with new pharmacological strategies that can offset the weight rebound. That's kind of, I think, the holy grail or the ultimate ambition for us, is to come up with therapies that when you stop to take them, you don't see this dramatic weight regain. And then another area of work that we are excited about is to try and understand the biology and the physiology of this upper intervention point. How is it that the body can correct against this overfeeding-induced weight gain? So there's quite little known about the homeostatic correction on this end, whereas much more is studied in terms of weight loss mechanisms in the weight rebound phase. So in terms of overfeeding, we've established this overfeeding model in rodents, where we use intragastric implantation of a catheter, and then we can chronically infuse high caloric liquid diet into the stomach of the animals. So unconsciously, they get a lot of calories into the stomach, right? And then we see we can make them gain 25-30% body weight over a period of 2 weeks. And again, like the human studies, right? Once we start to infuse water instead of liquid diet, they beautifully recover back to the controls again, showing that this system is intact also in this case, mouse models. And again, what's evident from our studies is that once we overfeed them, their voluntary consumption of diet completely disappears. So the body learns that they're getting a lot of energy, and the appetite is completely shut down, right? So they don't eat any chow. They have access to food throughout these studies. And then what's really exciting is this prolonged period of under-eating until they correct to baseline. So what is the biological mechanisms that are telling the brains of these animals to eat a certain amount of calories kind of below a normal energy consumption of a rodent for a period of time until they recover in body weight? Why is it how do they know 1 week later when the overfeeding has stopped that they should eat 30% less of a normal mouse in terms of recover? And we kind of are working to try and uncover these type of signals, and we believe that there could be endocrine factors that we have yet to kind of uncover. And we're not the only ones thinking about this. So this was a great review article by American scientists from 2014, kind of introduced this idea of a quote-unquote missing link in weight homeostasis. And we did our own literature survey in 2020, where we also rationalized this potential unidentified hormonal factor or factors against overfeeding. And a lot of the kind of very exciting research that are supporting this idea of a circulating factor actually dates back to the 80s, where several studies were done, but some of them were with these parabiotic models, where you actually surgically connect the skin of 2 rodents, 2 mice or 2 rats, so they share blood circulation. And then if you overfeed partner A, then the response in partner B will tell you whether it's a circulating factor that is potentially transmitting the response. To see if you overfeed partner A and partner B gains fat mass, it's likely due to or reduces fat mass, or vice versa, it's likely due to a circulating factor. So some of these studies have contributed to rationalize this existence of hormonal factors driving this response to overfeeding. So what we're doing now is a little bit kind of more modern approach. We're using these rodent models, and then we're kind of agnostic to what tissue we're looking at. And then we use multi-omics or other tools to try and study, could there be circulating factors encoded and secreted from, for example, the white adipose tissue or the liver, in terms of telling the brain that these animals are in in a hypercaloric state. And ultimately, we're interested in human health and human physiology, right? So we also embarked on a small human clinical overfeeding trial some years ago, and we're now kind of working to kind of make sense of all the data. But basically, we spent 1 week to try and find out what these subjects would require in terms of weight stability, energy requirements, and then we randomized them to go in the 50% overfeeding trial and stay in the control group, and then we took a lot of biological material. And just to show you a little bit of the results that we have yet to publish, so basically we can also make humans gain weight, not as much as the rodents once we overfeed them in the lab, but they also lose it again, as we've seen in the literature. This is just kilograms and percent-wise, so you see it's much less pronounced, obviously, than in the rodents, and there's also some variability, right? Because I now want to change a little bit more to talk specifically the last bit about the pharmacotherapies for weight management, right? And I think we don't need to go through the entire history of weight pharmacotherapies, but we're going to start around 2021 when we saw the current generation of incretin therapies being approved by the American authorities for treatment of obesity. And this is this once weekly version of the gut hormone GLP-1 that's been modified to bind albumin and they stay relatively long in circulation. And we saw in these phase 3 trials approximately 15% or so body weight loss in humans with BMI above 30. And this has really kind of was the starting point of this field that has been really, really rapidly evolving over just the last 5 years. But it was clear even before 2021 that targeting more than one biological pathway is likely superior, like in many disease areas, than targeting a single one. So maybe we want to target something beyond a GLP-1 receptor agonism, right? So in 2019, this just also, it was kind of speculated before that. This is just one example, but I think key from this paper is to show that already then and before this, it was shown in preclinical models that you can take peptides that have sequence similarity, and then you can integrate or mix agonism, so create single molecules that will then promiscuously target more than one receptor, right? So single peptide that activates 2 or more receptors, and that you have the birth of these dual-acting peptide molecules that have now also entered the market. Because I think so. Just to move back, so I think I can show it on the next slide. So the most progressed one is, of course, Eli Lilly that has pushed the tirzepatide, which is a dual GLP-1 and another gut hormone called GIP. So it's a dual peptide incretin hormone that has gone through clinical development and is in 2023 was approved for treatment of obesity. So when you kind of publish these simple overviews of some of these therapies, it's already outdated because the field is moving so fast. So this is one overview we published last year, but basically it's to show we have kind of semaglutide and then also a relatively new family of compounds based on amylin biology or physiology. And this is one of the analogs from Novo Nordisk called cagrilintide. It's also in phase 3. And then some of the older small molecule combinations. Then we have tirzepatide, which is this dual incretin molecule from Eli Lilly. And then we have Novo's combination of cagri and sema, cagri-sema. So it's a loose combination. It's not a single molecule. And then we have retatrutide from Eli Lilly. It's a triple-acting molecule that targets also the glucagon receptor in addition to the 2 incretin receptors. So it's one peptide that targets 3 receptor systems. And actually the phase 3 data shows it gets 26% or so weight loss now. So it's really kind of in pole position in terms of weight loss magnitude. What we're interested in, and I hopefully also some of you, is what are the central mechanisms that are mediating the appetite suppressing and behavioral effects of these molecules. And I think it's clear that more or less all the pharmacotherapies we're looking at today for weight management, they enter the brain through 2 sites, and that is either the hypothalamus, nuclei in the hypothalamus, or the brainstem, most notably the area postrema, and maybe to a lesser degree the nucleus of the solitary tract. So these regions that are relatively accessible for blood circulation, they express all of the receptors for these peptides, and that's how they kind of affect human behavior ultimately, right? So a big kind of question mark is what is actually the central pathways and circuits that transmit these signals into altered behavior, because it's complex behaviors, human eating, right? Eventually, it's also related to motivation, kind of memory, cognition, and all kinds of other complex behaviors that these drugs are affecting, right? And we also see a lot of inter-individual variability in how people are responding to these type of therapies. So we continue to go back to this kind of weight regain. This is something that we in the lab believe is very important to both understand, but also kind of try and develop drugs to offset this. So one way to go about this is to again dive into the neurocircuitry of these different hormonal or neuroendocrine regulators of weight regain. And one way is to kind of home in on the primary hunger encoding neuron in the hypothalamus, called these so-called AGRP neurons. And then you can start to kind of decipher what are the different circuits that have input onto the AGRP neurons, what hormones do they see from the periphery, and try and uncover, are there any kind of new molecular targets or pathways in this circuit that we can intervene with respect to pharmacology. So I think that's kind of a more modern approach, right? That we try and really understand all aspects of these key neurons that are involved in appetite control. And one hormone that kind of emerged over recent years is called LEAP2, and it's a hormone that it's an endogenous hormone that antagonizes the hunger-promoting receptor ghrelin receptor, and we've also worked on this, both trying to understand, because it's kind of relatively new in the space. It's always exciting to see its role. So we've both worked in the physiological control of this, and also recently published a paper. I will just show, we got the cover of we were happy about this kind of a cartoon cover, where we showed that if you use a LEAP2 analog together with a GLP-1 analog, you see potential offsetting of the weight regain trajectory. But ultimately, I think we really need to look beyond these kind of traditional pathways if we want to come up with innovative ways to sustain weight loss also after we've stopped treatment, right? So our journey into this has been to look into some of the modulators of neuroplasticity, more canonical pathways of the brain, such as the glutamatergic neurotransmission, and also based on genetic studies that have confirmed that variability in body weight have genetic aspects that's linked to NMDA receptor-related neuroplasticity. So we started in the lab to work on pharmacologically targeting the NMDA receptor for weight loss, and with the idea of potentially modulating the neurocircuits of appetite in a more sustained fashion, and not only targeting with incretin drugs. So one way, because the tools we have for targeting many ion channels in the brain are small molecules, and they have some benefits, but one of the downsides is they typically have access to the whole brain, right? And if you want to target appetite circuits in very distinct areas, you don't want these drugs to target all over the brain. So in order to mitigate this, we kind of took advantage of this Trojan horse approach in which you use a chemical linker to attach a small molecule to a peptide, and then you use the selectivity of the peptide and its specific peptide receptor to introduce a small molecule to cells of interest, right? So this is one way of using basically the biological tools we have from evolution to target small molecules a little bit more specifically. So what we did in this case was to take gut peptides such as GLP-1 or GIP or combination, and then attach with a chemical linker small molecule NMDA receptor channel blockers with the ambition of trying to deliver selectively these channel blockers to only regions that expresses the peptide hormone, such as the hypothalamus and the brainstem, and thereby avoid whole brain exposure of the small molecules. And this published now a couple of years ago, when we could basically show this small molecule channel blockers, GLP-1, that at a very low dose in terms of small molecule, where we don't see anything, that we see a high potentiation once we attach the small molecule to the GLP-1 peptide, and to a lesser extent if we do a loose combination. So it seems as if the targeted delivery is important in this case in the rodent model for potentiating weight loss efficacy. And again, did a ton of studies of this, but again, just one way of showing that we don't have this off-targeting aspect. So we there's an open field paradigm where you just inject the rodent, you put it into a box, and then you video record its behavior over a certain period of time. And you can see if you inject this small molecule, it runs around like crazy. It gets hyperlocomotion. But if we put the small molecule onto the peptide, we see that it acts like the control animals or the peptide animals. So it's just one way of showing that we circumvent some of the adverse effects of the small molecule if we attach it onto the peptide. So this kind of started already in 22 when we were working on the manuscript, also kind of spin-out adventure for us. So together with two members from my lab, we formed a spin-out company called Ousia Pharma, and I've taken this picture because these are the two co-founders, Jonas and Anders. And then the third guy in this picture, any of you know who this is? I don't expect you to know. Maybe if you, yeah. Okay, fit. Yeah, it's a Danish king. Very good. So you don't get better kind of commercial exposure than this, right? So we were incubating in this Bioinnovation Institute in Copenhagen, and he was kind of on a like this one-day visit, and they got a good picture of him standing with our company logo. So we're very happy about this. It's nice exposure. No, but then I think for us, we got a lot of this kind of smaller, non-dilutive funding, but ultimately we managed to get a venture capital investment, and you can say good and bad things about venture capital, but I think at least it's one issue as a kind of academic startup. It's one way of at least testing your idea to see if it has commercial legs, right? So now we're working with an American venture fund to see if we can kind of advance these therapies based on these peptide small molecule conjugates into clinical development. So I think that could be one new avenue for obesity therapeutics, at least we think so. But then the final part, and then hopefully we still have time for some discussion, is beyond the cardiometabolic disease space. So we're very interested in the applicability or the potential for these quote-unquote weight loss or metabolic drugs in their effects as neuroprotective agents. So their potential for treatment of, for example, Parkinson's, Alzheimer's disease, and beyond. So I think in order to make significant progress in this space, we really need to better understand how these agents potentially affect conditions beyond appetite and weight loss, because that can guide the development of more targeted therapies. I think the current therapies have been developed to treat obesity and diabetes, but if we want to kind of reuse these therapies for targeting other CNS indications, I think we can think about ways of maybe improving their brain penetrance. Maybe it's not enough. They only get to these sites surrounding the third and the fourth ventricle. Maybe they need to get deeper into the brain. How do we leverage their entry into the brain? That's one way of thinking about it. Maybe we need to combine with something else than just kind of more metabolic medicines in order to get potential benefits. Because there is a ton of preclinical data supporting that these gut peptides, they could have neuroprotective actions, although there's also recent clinical data suggesting that the potential is maybe not as high as initially dreamed about. What we've been doing is preclinical, also trying to test some of these well-known drugs in models of Alzheimer's disease. So this is unpublished work to just show where we're moving as a lab. So we're testing these molecules in models of Alzheimer's disease, just weight curves and some immunohistochemistry. And then what we're doing is to kind of set up understanding of their efficacy in order, and then we test our own kind of next generation peptide drug conjugates that have been designed to modulate, for example, neuroplasticity or neuroinflammation or other aspects of neurobiology in terms of their efficacy in first case in animal models of for example, neurodegenerative disease. So with that, I've reached the end. I just want to quickly run through it. So I think a key message is, I think it's clear that there's bidirectional defense of body weight, so homeostatic mechanisms that kind of defend body weight against major fluctuations in both directions. And it seems as if we haven't touched too much upon this, but in obesity, it seems as these homeostatic mechanisms, they actually largely persist and are not dysfunctional. And the current therapies for weight homeostasis, they act predominantly through brain circuits. And now, again, we see now, I think, a shift in the field going from kind of quantity of weight loss to quality of weight loss, whatever that is. And then finally, I think it's exciting that many of the circuits in the brain that we think about when we talk about energy homeostasis and food reward, they overlap with circuits related to other CNS physiology, but also pathophysiology, and kind of the implication in terms of drug development in this space is really exciting. So with that, I want to again acknowledge the lab. This is not the lab. They just didn't come the day we had to take the picture. This is kind of the current lab size-ish from my group and all our funders. And again, I really want to thank you for the invitation, and I look forward to hopefully a good discussion. Thank you again.
Thank you so much. So I'm going to call you Chris now.
Please.
Yeah. So for a very inspiring and very informative, amazing seminar. I'm sure there are a lot of questions from the ground. So please start while we are arranging the things. Yeah, just raise your hand and then, okay, please, Haojie.
My guess is, this conjugate can recognize both the receptors, right? And I was wondering, have you tested whether this conjugate can affect their binding affinity or the downstream signaling activation efficacy of each receptor compared to the peptide alone or the antagonist alone?
The short answer is no, we haven't tested. And you can easily imagine that if you kind of influence the molecule in whatever way, it can affect both its binding and its signaling properties at the GPCR, can affect the pharmacokinetic properties of the drug, all kinds of other aspects that could actually drive some of the effect you see. And that's something, again, we're trying to find out. I think we've tried to exclude that it's not, for example, pharmacokinetic properties. That's something we start with. So we just haven't created like a longer half-life GLP-1. We've tried in the paper we published, which showed with some slice electrophysiology that we have NMDA receptor blockage. So at least there is something, and we've used indirect means also to demonstrate this, for example, with transcriptomics and proteomics. But it's a little bit black box for us, some of the molecular mechanisms at this point.
Thank you. Hi, Chris. Great talk. Thanks for giving us such a great overview of all the things happening. I wanted to ask you one question about the first part, about the bidirectional aspect, and maybe you can elaborate a bit more. You were speaking about it earlier, that there is the set point, two of them. You think about like the upper and the lower. And is there any thoughts about when these set points get determined in individuals, and how does it change? Like, is it a permanent set point, or what about range?
Yeah. So I think I didn't get to elaborate because it's very speculative, and it's really tough sometimes to reconcile why we have obesity if there are homeostatic regulators of body weight. So I think the way we talk and think about it in the lab, because we do this, we think about it a lot, is we believe that there is a weight trajectory. So, and that's likely determined through first genetic. We know from family and twin studies that genetic variability or variability of body weight can be explained like between 40 and 70% by genetics, right? So whatever genes you inherit will determine your weight curve. And then I think there are critical periods of life, for example, preconception, in the womb, post-birth, and then some early periods of life, growth spurt, puberty, where I think that trajectory can be pushed in either direction depending on the environmental triggers, right? But then I think once we hit adulthood, there's a relatively generalized, relatively set body weight trajectory that you're on, and that trajectory can be difficult to deviate from. And it's between that trajectory then have each upper and lower intervention point. So you maybe have a genetic predisposition and you may be on a trajectory towards 130 kilograms in the environment you're living in. It doesn't mean you don't have the homeostatic regulators. They're just operating around that trajectory. I think that's how we think about it at the moment, and that's also how you can reconcile the state of obesity with still having an upper intervention point. I don't know if that holds true, but that's one model.
Yes, thank you for the presentations. I just have one question, and also really nice that I saw you talk a lot about the impact of brain axis, like brain gut or brain pancreas. So one of my question is that, do you think that targeting the enteric nervous system would also be a nice alternative approach to approach obesity? Because I guess enteric neuron does have the ability to sense nutrient, and they also interconnected with the vagus and the brainstem and the whole brain compartment. So do you think that it's a better druggable target compared to the brain because you don't have to cross the blood-brain barrier? Also, what do you think about the shift toward maybe more of the brain-gut axis in treating obesity, for example?
Yeah, it's a very good point. And I think actually the field has been a little bit kind of misled for some 10 years or so initially, right? Because you have these receptors in the brain, and then we predict or assume that a lot of the effect is driven directly by the brain as opposed to local signaling that's then transmitted to the brain. And I think it's clear that the physiological effect of many of the gut hormones must be locally regulated because of their short half-life in circulation. So it's relatively unlikely that the endogenous GLP-1 actually acts as a central appetite regulator. And how to leverage that therapeutically, I don't know, but I don't see, why not? I think it's fair to assume there could be a future for both a peripheral to central circuit as well as a direct central circuit. I think there's relatively strong evidence to show that some of the long-acting gut peptides, they actually mostly work through the central nervous system through loss of function models. Yes, it's a good point. So what is the role in general of epigenetic biology in weight homeostasis? I think it's kind of, the jury is still out, right? There's a lot of association-related studies also in humans that shows, again, in different stages of even conception, etc., that you have epigenetic markers that are being regulated. It's tough to, I think, demonstrate kind of causality in terms of direct implication. But I think, yeah, why not? I think we should be open to also epigenetic contribution to the modern obesity epidemic, because it's happening so fast, right? That I think obviously our genomes are not being regulated, but it could be the way the genomes are being basically being transcribed or being basically assessed, right? So I think it's a little bit of a vague answer, and I think there hasn't been too much also in terms of looking at pharmacology and epigenetic control. So I think there's definitely also some more work to be done in terms of understanding this. Thanks.
Hi. So you mentioned homeostatic circuit, right? In the control. So what about next circuit, right? So how does that feature in the management of obesity?
Yeah, very good question. So I think for many years, at least, there's two things, if we have time, I want to say. So there's what is the role of hedonic or feeding or food reward in obesity pathogenesis, and what is its role in kind of pharmacological treatment? So I think it's clear that this midbrain reward circuitry plays a major role in our feeding behavior. And I think that's also kind of an intuitive go-to explanation for why is it that we end up in positive energy balance on a day-to-day basis, right? So anytime during the day, you're being bombarded with kind of stimulating foods, right? And it's easy to, and many of them are energy dense, so it's easy to overconsume in a temporal fashion, right? And then we rely on our homeostatic control to correct this on a kind of longer time scale. But whether that's actually always 100% corrected is I guess one explanation for this. A lot of these scientists, they adhere to this idea of that these hedonic or food reward circuits are major drivers of modern obesity epidemic, right? So that's one angle. And then I think for the pharmacotherapies, it's clear that they indirectly influence those circuits. So you can see if you give an incretin drug, you can see changes in dopaminergic signaling in the nucleus accumbens, for example. So there's clear effect. It's likely not direct, but it's likely through feed-forward circuits that comes from the brainstem and then ultimately it hits this midbrain circuit. But there's clearly an effect on hedonic aspects with these weight loss therapies. That's also why they're being trialed and have demonstrated preclinical and some clinical effect on, for example, substance abuse, such as treatment of alcohol addiction or smoking addiction. There's also clinical studies ongoing to evaluate these weight loss drugs for these other indications.
I'll take a second question if somebody else doesn't have a question. I'll just wait for a minute. Yeah. Okay. So my question is again related to what you're saying. So among the the different drugs you described, one of them was naltrexone and bupropion, which was also being used as a potential anti-obesity drug, right? So are the trials now trying to combine the two, because normally that was used for a addiction response, right? So do you have more comments on that, like combining the completely different type of agonists, right?
No, but I think so. So those small molecule combinations are largely out of fashion now for obesity, for weight loss management. But why not consider a low dose of the GLP-1 therapies with some of these small molecules for treatment of? And I think that was my point in the end, right? That if we want to kind of be inspired by this class of weight loss therapies for treatment of other CNS indications, such as addiction or whatever, we can. I think there's many ways to consider combining with already repurposing of existing therapies. And we haven't, because we haven't fully understood if there's potential additive or even synergistic effects sometimes by combining these type of drugs. So I think, there's still a lot of room for studying the combination of new drugs with older drugs, if you like.
Actually, I have a question. Can you elaborate about the quantity and quality?
Yeah. So I think I'm trying to follow a little bit what the companies are doing, right? So they've now really they've been fighting for this, who can get the most weight loss in a phase 3 trial, and that's basically seems to determine their stock price. But I think now it's clear they all have oral programs. I didn't talk about that, but going from injectables to oral therapies seems to be really a major focus now. So Eli Lilly has a small molecule GLP-1 that seems to be very interesting, and Novo has maybe a little bit weirdly kind of decided to develop a way in which they can introduce oral peptide without kind of still have some bioavailability. They lose a lot of drug, of course, but they've now entered the American market with the oral GLP-1 also. So, and those oral versions, they don't give the same weight loss, but of course they're much more convenient for the consumer. And ultimately, small molecule may eventually be easier to produce and distribute. It doesn't have the same requirements have to be at the temperature control conditions, etc. So that's of course not quality of weight loss, but that's then moving towards that is of course looking at all other outcomes. So I think a major breakthrough some years ago was the cardiovascular outcome trials, where they found that GLP-1 therapies also have benefits on cardiovascular outcomes, because historically many weight loss drugs would have negative effects on cardiovascular outcomes. But now you see like this list of benefits on fatty liver disease, chronic kidney disease, even some skin disorders, osteoarthritis, etc. And you see a growing list of benefits of these drugs. And that's, again, goes to like, what is it actually we're treating? Are we treating a percent of body weight, or are we actually wanting to treat a range of comorbidities? And that goes more towards actually the quality. And then finally, the major downside with the current therapies is tolerability. So adverse effects pertaining to nausea. So mitigating that is also going to be a potential game changer if you can come with a therapy without losing efficacy, but you kind of have less of the nausea aspect. So that's also, so those are kind of the more nuances today, and it's not only about getting the most weight loss.
So I am tasked to ask a few questions which engage especially the junior scientists, right? So like, as you can see, your research spanning from biology, application of obesity, metabolic disease, and now brain disorders in pharmacology. So what advice would you give for persons working in interdisciplinary fields?
Yeah. No, I think so. I was fortunate during my postdoc to be introduced to an environment in which hardcore peptide chemists were working with physicians and biologists. And I saw that this could actually, although they didn't speak the same language because the biologists didn't understand chemistry, they still made it work. And I think for me, that was very inspiring. So when I moved back to set up my own lab, I just tried to emulate that. So I immediately went to small molecule chemists, peptide chemists, and said, we need to work together on this. And we managed to find students that could be the main kind of integrator between these two fields. So over the years, I've learned a little bit more chemistry, not a lot, and vice versa. I would like to think that the chemists have also been more kind of exposed to the biology. So that's just one way of interdisciplinarity that I think we've been somewhat successful with. And then, of course, now moving to the era of multi-omics, big data, AI, it's also a whole new level of expertise, right? And some of us kind of more older scientists, we don't have the same training in these areas, right? But I think if we want to stay at the forefront of our field, we have to really try to adapt. And so I think that's just a part of it. To be open and then also trust. Sometimes it's a matter of trust in terms of collaborators that if you have a gut feeling or you feel there's a personal connection, then sometimes trust in terms of research interaction and collaboration.
I like the gut feeling part. Yeah. So we have a lot of information. We also have little time. Also, given the field is moving so fast. Yeah. So how would a young clinician, scientist, or basic scientist, basically hone their skill sets and then keep up to date, right? You get just now you basically say, okay, finding collaborators. But as a young researcher, right, it's just started. Any advice for them?
In terms of keeping up to date with it?
Yes, and then finding their way and try to think about how can they become you later on.
I don't know if I want to recommend that. It's very stressful. No. Yeah, it's tough, right? Because even, and I'm not that old, but even when I was a student, right, you'd go onto journal web pages once a week because that was when the new papers were released, and then you kind of had like a schedule where you could follow the literature. And then you come social media, right? And there was Twitter, and it was great for a few years. Then come X, and then it's kind of downfall. But it's so today can be so overwhelming to keep up with the literature, right? Because there's just published thousands of papers all the time. So I think I don't know how to deal with it. Just accept that's how it is. And of course, you find your own niche, and you try and follow the development within that. But sometimes you also, I think you have to, again, trust the direction that if you set together with your PI for what you're working on. And then don't always try and kind of be worried too much about what everyone else is publishing or reporting at the same time. Of course, you have to be aware of it within your own field, but otherwise you can easily kind of be discouraged or drown in all the information that's constantly being bombarded with.
That's really good advice. Any other burning question from you, please?
Yeah, thank you a lot for your lecture. And I have a question for you. Do patients develop a pharmacological tolerance to weight loss medications like GLP-1 agonists, as well as body attempts to restore its metabolic set point, and how it can be addressed clinically?
Yeah, no, very good points, both of them. It seems as if that there is very little development of insensitivity or tolerability in terms of the weight loss drugs. So there are trials with 4 years on treatment where there's no rebound. So there's now clinical trials where people have been on the drugs for 4 years and there's no rebound. But I still think that there's a lot of quote unquote non-responders also, and there's a lot of individual variability. So I think there's still a huge need to have a broader variety of weight loss drugs so you can kind of substitute treatment. And I think, again, something with the future in terms of weight loss treatment is we'll see many of the current therapies will be used to drive weight loss down, and then maybe some more tolerable treatments can be used to sustain that weight loss. So I think weight loss and weight loss maintenance could be treated as two separate entities. I think that's an interesting way, at least I think, to consider this. And then this set point adjustment, I think so far there's no evidence that this is possible except for the bariatric surgeries. But I guess you can argue that that's also it's also somehow a chronic intervention, right? But there's no, at this point, that speaks to that we can readjust our set point, especially not to the lower state. But I think that's what our ultimate dream is, to kind of rewire the brain so it defends a lower body fat mass in the end, right? But it's still kind of it's a kind of lofty or ambitious dream, and that we have no data to really support this yet.
Okay. One last call for question from the audience. Oh, okay, great. Please mic the
You mentioned the differential responses, the inter-individual differences. So how does the timing of onset of obesity influence this? So does early onset, particularly childhood obesity, do they respond differentially from adult onset obesity? And also one question on how does adipose tissue morphology play into this? So people having a more hypertrophic or hyperplastic morphology, do they respond differently to these drugs?
Yeah, so great questions. And this is now, again, both what the company is doing and the basic researchers or clinicians, we're trying to find out why does people respond differently. So now there's a big push towards precision medicine, obviously, right? So one approach is deep phenotyping. So figure out what is the clinical phenotype of patients, how do we subdivide them? And then also using, let's say, proteomics to see are there biomarkers that can predict what class you belong in, and then kind of reverse translating this into to try and come up with ways of subclassifying individuals with obesity into if you can predict responders from non-responders, then ideally you develop new therapeutics for the quote-unquote non-responders, right? But it's a huge topic at the moment to try and understand, because obviously it's big business if you can understand why people don't respond also to the current drugs. It's a very exciting area.
Okay, so one last question. Please give 1 minute golden advice to the audience.
I'll try. This is the worst part of it. It's because you have to really think. No, I thought about two things, and I almost said both of them, but I'll say them again because they're important. So I think first one is more generic, and it goes back to what we just talked about. So I think in today's world, as a researcher, you can keep yourself busy with generating data all the time. So we're just like you work a lot here. You can just continue to generate data, and you have so much data to handle and deal with all the time that it can kind of maybe somehow deflect, or at least I feel it can move me away from thinking about why was I doing this in the first place. So what was my initial mission for going into research? What actually excites me? What are my motivation? And then somehow years go by, you just continue to work, right? And then I think it's important to keep track on what your initial direction was and why you set out to be a researcher in the first place. And sometimes just kind of keeping ourselves busy can sometimes distort, I think, the direction. So I think while we're being very busy, and it's very exciting when we do, I think we need to somehow go back and reflect about if we're on the right course, if the initial reasons for getting us into this topic that we're now working on, if those are still kind of aligned with what we do. So I think that's kind of one generic one, maybe also more than 1 minute now. And then on obesity, I really think it's still important that we acknowledge obesity and overweight as a chronic relapsing condition, and we accept that it's a chronic condition next to many other modern diseases, and we don't consider it as a failure of poor lifestyle or poor willpower or self-control, which is still a case, and it contributes to stigmatize individuals with a lot of body weight. No, the fact is that no one really wants to have obesity, and people have typically tried all kinds of diets to get rid of it. So I think we also need to treat obesity with kind of more respect as a chronic condition. And what we've gone through is that there's a huge genetic predisposition, and there's an environment that triggers some individuals to develop excess body weight, and we have to kind of help that by changing the environment, or perhaps at least in the meantime come up with good therapeutics. So those were the golden advice.
Thank you very much.
Yeah.
So now let me invite Prof Time from the chair of the council to actually pass a token of appreciation from the NUHS community. And Prof Time.
Thank you.
So thank you. So once again, let's thank our speaker, Professor Clemmensen. Amazing one. And thank you all for being here.